pleasant day to everyone, especially to our distinguished panel members. I am Charles Guillermo, and on behalf of my co-proponents, we are pleased to present our study entitled In Silico Functional Diversity Analysis of Soil Bacterial Metagenomes in Agroecosystem in Los Baños, Laguna, Philippines in comparison with karst ecosystem in Taft, Samar, Philippines. Today's presentation will revolve on chapters flashed on the screens. Now we begin the presentation with bacteria. Bacteria is a major soil inhabitant known to represent the vast majority of the overall below-ground cellular biodiversities worldwide and is known to play a crucial role in the functional aspect of ecosystems in global biogeochemical cycling, contributing to soil structural and functional assets. Understanding the functional diversity contributes to better comprehension of specific roles soil microbial organisms play in the ecosystem. Despite its recognizable progress in microbial research, greater efforts are still required in the Philippines. This led us to assess different ecosystems vitally present in the country. The rhizosphere is a prominent hotspot of soil activity and is home to abundant populations of microbes, which is dominated by bacteria. As reports mention agricultural lands being a major landform, and with rice being a staple food in the Philippines, sustainable production is yet to be uncovered. As a contribution to crop output increase, we should shed light on the functional diversity of rhizosphere-dwelling microbes. A significant yet fragile ecosystem is the forest over limestone, or simply karst forests, with an estimated 11.7% of local land cover. It is endemic to rich kinds of floral and faunal species as well as abundant microbial diversity. Scarcities in the characterization of bacterial profiles are observed, and studying this can aid the conservation and monitoring efforts of the landscapes. Now, this study with the above-mentioned motivation and causes shall gear its attention to characterizing soil bacterial functional diversity in both rhizosphere in agroecosystem and forest over limestone karst ecosystem by thoroughly investigating the functional traits of bacteria and their effects to the ecosystem, elucidating similarities and differences in contrast with karsts being a minor landform and agroecosystem being a major landforms in the country's vast land cover. To achieve this, a relatively new, rapid, culture-independent technology featuring a high throughput yield of results shall be the prime approach in this study. Interestingly, a revolutionary bioinformatics tool called MGRAST can be used to annotate functional profiles of microbiomes. This strategy allows researchers to comprehensively expand knowledge on soil inhabitants across ecosystems. Now, the study's research paradigm involves data mining procedures through MGRAST server, applying category filters to access available raw data for metagenomic analysis, which in turn provides valuable information on the soil's bacterial functional diversity thriving in the two sites of study. The statement of the problem revolves on four key main aspects. The Philippines remains to have environmental sustainability issues, and most studies focus on taxonomic, floral, and faunal microbial characterization. Hence, microbial functional roles in ecosystems need more attention. Bacteria playing significant roles in the ecosystem means that their ecological importance must be deeply understood. With the functional diversity of bacteria known to impact the stability of ecological transects, data will be useful in achieving ecosystem sustainability. With all of these at hand, our study shall determine in silico metagenomic profiles of soil microbial communities in rhizosphere in the agroecosystem in Los Baños, Laguna, using MGRAST software and compare it with the existing karst ecosystem bacterial functional profiles in Taft, Eastern Samar. For the selected points of study, only the functional profiles specifically from Los Baños, Laguna and existing data from Samar will be characterized. Moreover, the analysis will only be limited to KEGG orthologies levels 1 and 2 for bacteria. The outcomes of the study shall directly benefit two main sectors: the policymakers involving the local government units of Samar and Laguna, the DENR and Protected Area Management Board. The academic value of this research shall also benefit the biological research departments of universities, including DLSUD, and especially the researchers. The imparted knowledge shall aid in their pursuit of knowledge and scientific-based conservation practices. Now, let me hand the presentation over to Ms. Layson.
Thank you, Mr. Guillermo, and good afternoon to our panel members. Allow me to present Chapter 3, Methodology, which consists of the research design, research setting, and research procedure. The research is qualitative in nature to provide a more in-depth and wide understanding of bacterial communities present in the mine data. The method is designed to mine data from already established databases, so the paper will utilize an in silico descriptive approach using bioinformatics and visualization tools to effectively characterize the functional profiles between agroecosystem and karst ecosystem. The raw data mined from the MGRAST server is based in Los Baños, Laguna as the representative data for agroecosystem, particularly within the rice paddies of International Rice Research Institute in the Philippines. On the other hand, the representative data for the karst ecosystem is based in Taft, Eastern Samar, which is one of the municipalities covered by the SNIP, or Samar Island Natural Park. Processing of these raw data is presented in the diagram in Figure 3. This summarizes the research methodology. The collection of raw data will be followed by pre-processing, then the assembly of metagenomes, and lastly, data gathering to generate an analysis profile to visualize the functional profiles. For the raw data collection, the files were first received as a FASTQ file, a format compatible with MGRAST, and were uploaded to the pipeline for pre-processing. This is where low-quality regions are trimmed and paired ends are identified and joined. Processed FASTQ files will be dereplicated to remove artificial duplicate reads, or ADRs. These ADRs will undergo quality assessment to determine sequence quality. After quality assessment is established, sequences are screened, so reads that do not match the model organisms are annotated. Fragmented genes are detected by gene calling, then the relative abundance is maintained by clustering of sequences formed at a 90% sequence similarity level. A representative from each cluster is selected for representational similarity analysis. These representative species are functionally identified to detect sequences that match a protein or DNA sequence. Sequence similarity research will be carried out via M5NR, a non-redundant protein database, to generate information from different databases such as RefSeq for taxonomic data and COG, NOG, KO, and SEED for functional data. The data are visualized using tools available in MGRAST, and this ends the methodology. I will now discuss the results and discussion in Chapter 4 along with Mr. Salgado. The data to be presented will comprise of number 1, results for agroecosystem, as well as number 2, its comparison with the results from karst ecosystem, which are adopted from the visualization tools via MGRAST server. We utilized KEGG orthology database analysis to characterize the functional profiles in both ecosystems for domain bacteria. Allow me to address the first objective. Presented is Figure A, the pie chart of the annotated functional profiles of bacterial communities in rice rhizosphere and agroecosystem under general functions, or level 1, which will be followed by level 2 later on. The KO database is represented by 6 functional categories, namely metabolism, environmental processing, genetic information processing, cellular processes, human diseases, and lastly, organismal systems. In this chart, metabolism gets the greatest portion with 57%. Hence, we can observe that dominance of metabolism-related genes in agroecosystem, as well as information processing and cellular processes, are dominant. And according to literature, these functions are greatly significant in maintaining the basic metabolic activities of soil microorganisms. The next presented data, which is in Figure B, is the annotated bacterial functional profiles in the rice rhizosphere and agroecosystem under level 2. The annotation revealed richness in amino acid metabolism by 19%, carbohydrate metabolism by 12%, and membrane transport by 9%. Other functional categories annotated were translation, metabolism of cofactors and vitamins, and energy metabolism for both ecosystems. Both level 1 and 2 results will be compared with the bacterial functional profiles in a more specific manner for the karst ecosystem by Mr. Salgado.
Thank you, Ms. Layson. Allow me to explain the second objective, the similarities and differences in the functional diversity of rice rhizosphere in agroecosystem and karst ecosystem. Starting off with our level 1 KO analysis, we take a look at the functional profiles of both rice rhizosphere in agroecosystem, shown by the inner circle of the donut chart, and the functional profile of karst ecosystem, shown by the outer circle. Here are our findings. The same proportions in the functional categories were observed across the two ecosystems. Genes for metabolism presented dominance as it takes a large share in the overall functional data hits. The functional trait for cellular processes stands out due to the distinct differences in the trait proportion between the two ecosystems. The functions environmental and genetic information, processing, human diseases, and organismal systems all exhibit the same similar functional trait proportions between the two ecosystems. No notable differences were identified for these four functions. Next, we observe our level 2 functional profiles, beginning with metabolism. As shown, the functional profile for both karst and agroecosystem are very similar, with the functional trait for amino acid metabolism being dominant for both ecosystems, followed closely by carbohydrate metabolism and energy metabolism. Our finding shows that the abundance of genes responsible for amino acid metabolism can be attributed to their purposes as an intermediary to a crucial cycle that bacteria partake in, the soil nitrogen cycle. Bacterial metabolism plays a key role in the karstification process involving the weathering of calcium carbonate present in the karst landscape. Bacterial metabolism is identified to support plant growth as plant growth promoting bacteria, PGPRs, such as nitrogen fixers and phosphate solubilizers. Next, we have our level 2 KO analysis for cellular processes. As we can see, this is the dataset that presented the largest difference between the two ecosystems, with cell motility being dominant in agroecosystem and cell growth and death being dominant in karst ecosystems. Our finding shows that the dominance of genes responsible for cell motility in agroecosystems highlights the role of bacteria as essential root colonizers. The moral property of bacteria provides aided superiority in acquiring nutrients, escaping from toxic matter, approaching the desired host, and colonizing preferred sites. High levels of genes responsible for cell growth and death in karst ecosystems can be linked to the abundance of available organic carbon sources and the role of bacteria in enzymatic weathering. The iterative process of cell growth, generation, and death all collectively take part in reforming biological components. The following datasets will show extreme similarities in functional proportions of the two metagenomes, and thus will be subject to more in-depth analysis in future studies. Moving on to environmental information processing, we can see that the function proportion is very similar between the two ecosystems, with genes for membrane transport being the most dominant, followed closely by signal transduction. The functional profile of genetic information processing for both ecosystems are once again very familiar, with translation being the most dominant, followed by replication and repair, folding and degradation, and lastly, transcription. For human diseases, representative hits for cancer genes are the most dominant, followed closely by infectious diseases and substance dependence. Lastly, for organismal systems, environmental adaptation is overwhelmingly the most prominent functional trait. Our findings show that little to no representative hits for signaling molecules and interactions were observed for both karst and agroecosystems. The dominant presence of cancer functional genes in both ecosystems does not necessarily mean that bacteria are causative agents. Functional orthodogs belonging to disease pathways were just identified. The predominance of the environmental adaptation trait for both ecosystems may indicate the ability of bacteria to rapidly respond to intracellular and environmental changes in order to survive. Due to the similarity of the data, level 3 functional analysis is required to thoroughly establish evidence for any claims. This is currently outside the scope of our study. Now, to discuss the conclusion is Mr. Guillermo.
Thank you, Mr. Salgado. Now we move over to the conclusions. Primarily, results revealed distinct patterns in soil bacterial functional diversity. Significantly, the metabolism illustrated relative abundance by most hits represented in both agroecosystem and karst ecosystem. This can infer its major role in the karstification and enzymatic weathering processes in karst landscapes and the nutritional influence of rhizosphere in rice-rhizosphere agroecosystems. Distinct differences are also observed, one of which is the genes responsible for cell motility with a more relative abundance in the rice rhizosphere in agroecosystem than in the karst forest ecosystem. This observable difference in predominance of genes for motility in agroecosystem is due to efficient root colonizing in rice rhizosphere. Similar metagenomic composition between two ecosystems are observed between environmental and genetic information processing, human diseases, and organismal systems. At the same time, low turnout of hits were constituted for human diseases and organismal systems. Thus, it is recommended to establish level 3 comparison on these functional traits to further characterize their differences or similarities. Now, as the results from this paper aims to contribute to the increasing pool of local and worldwide metagenomic function, data from this paper can be utilized by farmers and agricultural researchers in their pursuit of evidence-based ecological maintenance and manipulation according to microbial species thriving in the soil. This essentiality remains rooted to the prominent influence of bacterial communities in many ecosystems in the world, as well as the processes of microbial and ecological research being more accessible and convenient. Several recommendations are also placed for future researchers to take on. First is the inclusion of level 3 functions for a higher and more specific yield of results for comparison. Second is the conduct of metagenomic analysis of existing sequence files in order to discover more specific functions. Third is the conduct of metagenomic analysis once again to determine the thriving microbial communities in the rhizosphere, specifically in diseases and healthy crops. Fourth is the utilization of the metagenomic analysis of existing sequence files to discover functions such as antibiotic production and nutrient sources. Fifth is using the same way of analysis to determine the phylosphere's microbial communities in diseased and healthy plants. And lastly is the utilization of other databases such as COG, NOG, and seed subsystems. As we reach towards the end of our presentation, the scannable QR code link is flashed on your screen for easy access to the paper's compiled references. Now, I, Mr. Guillermo, alongside Ms. Layson and Mr. Salgado, would like to express our sincerest appreciation to the following: our thesis advisor and co-advisor, Sir Wilson Jacinto and Sir Edmerson Jeronimo, for their extensive technical assistance in the writing of this paper. Our subject professor for both Thesis 1 and 2 for their never-ending support throughout the run of the study. The UP lecturers which paved the way for us learning bioinformatics and metagenomics, and our respected panelists for giving us time for today's presentation. And that signals the end of our presentation. Thank you so much, and it is with great pleasure for us to be given this opportunity to present before all of you today.